All right, AP Bio. Good morning, afternoon, evening, whatever. So this is going to be the photosynthesis presentation. It's going to be kind of a quick overview, and so you can be able to use your notes. So what we're going to be looking at, we're going to be looking at photosynthesis, the light spectrum, chlorophyll, chromatography, action, absorption spectra, photosynthetic stages, and limiting factors. And so when we talk about photosynthesis, okay, let me get view page. Sorry about this. You're just going to have to deal with it. Single page. And I'm going to kind of sit right up here. So when we talk about photosynthesis again, it's a process by which certain cells synthesize carbon compounds. That's the key point. So for most often, they're going to say glucose, but you need to know, like often on the exam, they'll say carbon compounds from inorganics, so CO2 and H2O in the presence of sunlight. This process requires a pigment, right? And then the plants have specialized organelles adapted. And so if we look at the formula, we've got carbon dioxide plus water with the light coming in, right? Because it's an endergonic reaction, and chlorophyll will produce glucose, oxygen, and water. And so we talk about photosynthesis versus cell respiration. Photosynthetic organisms use light energy to create stored chemical energy. So the energy is stored in carbon compounds that's then released by cell respiration. So hopefully remember endergonic and exergonic. Animals cannot synthesize these compounds and then must consume them as food. And so that's kind of the key point there. So again, there's a visual. So that endergonic, exergonic idea. So when we talk about photosynthesis, we need to talk about the light spectrum. And so visible light from the sun is composed of different ranges of different wavelengths or colors. And so your visible spectrum is from about 400 nanometers to about 700. And so the colors of visible are from longest to shortest. So your reds are your longest, orange, yellow, green, blue, yellow, indigo, and violet. And hopefully you remember from biology, the reason why things look green is because green is being reflected, all that other light is being absorbed. And so what the chlorophyll is, that's the pigment that absorbs the light and converts it into ATP, intermediate or glucose, which is stored. Yes, plants do make a little bit of ATP, not as much in the chlorophyll, not as much as when you do cell respiration. So chlorophyll absorbs red and blue light most effectively, but green is reflected, hence the green color of the leaves. And there are some other accessory pigments, carotenoids, such as like your oranges and yellows. They may absorb other wavelengths to maximize photosynthesis, but it tends to be reds and blues. And so here's your action versus absorption. So if we look at this picture, the absorption spectrum shows wavelength. And so we got 400, 700, as you can see. If we get to the amount of light absorbed, if you get between that 5 and 600, right, not much is getting absorbed, right? If we look at the action spectrum, shows light wavelengths used by pigments, photosynthetic rate of photosynthesis, again, not so much between that 5 and 600, right? And if we go back, we can see between that 5 and 600 is your green. And so here are the stages. Photosynthesis occurs over two distinct stages. We have the light-dependent reactions, which convert light energy into chemical, and then the light-independent reactions, which use that energy. What was made in the light-dependent reactions are then used in the light-independent to synthesize carbon compounds. So we've got light water going in, light and water go in, oxygen comes out. There's your light-dependent reactions. You're producing ATP and NADPH. NADPH is a carrier molecule, and then I'll talk more about this in class. And then we go into our light-independent reactions. You have RUBP, GP, and TP. Essentially, those are just abbreviations for some compounds. It all occurs in the stroma, but ultimately what's going out is organic compounds, and then that's where the carbon dioxide goes in. It goes in during the light-independent reaction. And so the light-dependent reactions occur in the thylakoid, and light's absorbed by chlorophyll results in production of ATP. That's important. Light's absorbed by water, which is split to produce oxygen, hydrogen. You have to know what's going in, what's going out. And then the ATP and the hydrogen, which the hydrogen's tied into that NADPH, are used in the light-independent reaction. So oxygen is produced in photosynthesis from the photolysis of water. So there's your light energy, breaks up the water, right? That water's broken up, oxygen's a waste, hydrogen goes into the independent reaction, you get your chlorophyll, but everything going to the right is going to be used later. So you got chemical energy, and you've got NADPH and ATP. The light independent reactions occur within the stroma, so they are not regulated by light. So ATP and hydrogen are transferred to the stroma. There, the hydrogen will be combined to form a complex organic molecule, often we'll call this glucose when it's all said and done. And then the ATP provides energy required to power these anabolic reactions. Remember, anabolic, catabolic, so these are building. So these materials will diffuse in a chloroplast, right? You get carbon dioxide, NADPH, ATP. You got a series of these small metabolic reactions that are regulated by enzymes. We call that carbon fixation, and then ultimately you have a organic compound. And so there are some limiting factors, though, to photosynthesis. The law of limiting factors states that when a reaction depends on more than one condition, the rate of the reaction is limited by factor nearest its minimum value. So photosynthesis is dependent on a number of favorable conditions, including temperature, light intensity, and CO2 concentration. So the limiting factor sets the speed. If you got good temperature, good light, a lot of CO2, you're going to have a very quick reaction. You take away you drop the temperature, you drop the light intensity, you drop the amount of CO2, you're going to have a very slow reaction. So think of those three things as affecting the rate or the speed of the reaction. So temperature is a factor. Photosynthesis is controlled by enzymes, so rubisco. Increasing temperature will increase the kinetic energy of the enzyme, will increase collisions. At an optimal temperature, the rate will peak. However, if you get beyond that optimal temperature, you may start to denature those proteins, actually slow down. So if you've got the optimal temperature, you're going to increase the amount of enzymes binding substrate. It's going to be good. Light intensity, again, light is absorbed by chlorophyll and used to make ATP. As intensity increases, more chlorophyll becomes photoactivated, increase the rate. However, at a certain point, you're going to get a plateau. The activity will level off. And this is because all available chlorophyll are photoactivated and the rate cannot increase. So this looks like that enzyme one. And we talk about CO2 concentration. Carbon dioxide is involved in the fixation of carbon atoms to form organic molecules like glucose. And so more carbon dioxide means more carbons are made. After a certain point, though, the photosynthetic rate will plateau. This is because all the enzymes responsible are occupied. So when we talk about photosynthesis experiments, we can look at carbon dioxide uptake, oxygen production, and biomass changes. Again, we are probably going to be looking at oxygen production because we'll be able to see them float if we get to the lab. And so topic review, can you do the following? Write the equation for photosynthesis, describe the role of chlorophyll in plants, draw action absorption spectra, explain the two stages, identify different limiting factors, and list methods for measuring photosynthesis. Again, this was not the PowerPoint I wanted to do, but it's the one I have to do because we're kind of getting to the end of the before winter break, and I want to have a nice clean break. So anyways, I will do this in class. It's probably to your best benefit to go in and do these on your own, but you're going to put in what you're going to put in. So anyways, have a good day. I'll see you Monday or Tuesday, and this is me signing off from beautiful room 102 in Tiffin Columbian High School.